Welcome back. So, in this lecture, I am going to talk about enzymes and their inhibitors. So, I am going to talk about several mechanisms of enzyme inhibition, and I will also give you examples of several very important inhibitors. So, I will discuss types of inhibitors. So, there are reversible and irreversible inhibitors, and then I will type under reversible there are these major three different types of inhibition mechanism: competitive, uncompetitive and non-competitive, and then I will also give you examples of these different types of inhibitors. So, inhibitors are very important, and it turns out that most medicines that we take on a regular basis are inhibitors of different types of enzymes. So, not all medicines are inhibitors, but most of them are, and I have listed a few examples here. So, for example, angiotensin converting enzyme inhibitors are this captopril, enalapril, lisinopril, right. So, these are medicines which help relax the blood vessel and lower blood pressure. So, you will see that if, many elder people in your house, they will take some of these medicines for reducing their blood pressure. Then there are statins which inhibit HMG-CoA reductase. So, these are some of these medicines which reduce cholesterol level, right. And again, you will see many senior people taking one of these molecules on a regular basis. When we eat something very oily or something very rich, we might actually get acid reflux or acidity. So, in that case, we take medicines like omeprazole, right, and these other variants. So, these are proton pump inhibitors, and what they do is they reduce the acidity. So, they reduce this proton potassium ATPase enzyme, which is basically a proton pump, and thus they reduce the acidity of your stomach, right. Then there are these COX enzyme inhibitors. COX is cyclooxygenase oxygenase. So, these inhibitors are something that help us relieve pain, and these are some names you might have seen. These are very common: aspirin and ibuprofen, right. And then there are tyrosine kinase inhibitors. So, we have already seen the example of imatinib, right, and then there are other variants which are used to treat various kinds of cancer. So, I will also talk about some more inhibitors as I go through this lecture. Now, what are the types of inhibition? There are two types of inhibition. The first one is called reversible inhibition, which means that the inhibitor is not binding the enzyme with a covalent bond. Okay, so the interaction is reversible. So, we can increase the substrate concentration and maybe we can remove the inhibition, but again this is not always true. So, it turns out that there are three types of inhibitors. One is competitive, second one is uncompetitive, and the third one is non-competitive or mixed inhibition. So, in all of these cases, the inhibitor can actually diffuse out of the enzyme, right. Wherever it binds, it can bind, but it can also diffuse out. Second type of inhibitors are called irreversible inhibitors or irreversible inhibition. In this case, the enzyme actually forms a covalent bond and it binds it irreversibly. So, the enzyme activity cannot be removed or cannot be regained back, right. So, these inhibitors permanently inhibit the enzyme. So, let us look back at this. We have already seen this. So, this is our Michaelis-Menten curve, right, and what we have also seen is that this Michaelis-Menten equation we can linearize it, which was done by Lineweaver and Burke. So, what we do is we flip it, we inverse it, and then we divide these two by Vmax over S. So, this is what we get. So, 1 by V naught equals to Km by Vmax times 1 by S plus 1 by Vmax. So, this is in the form of y equals to mx plus c. Y is 1 by V naught, x is 1 by S, it is multiplied by m, which is the slope of the line, which is Km by Vmax, and a constant c is 1 by Vmax, which gives us the y-intercept. And when we plot 1 by V naught by 1 by S, it will look something like this. So, the actual data will be somewhere here, and then we can extrapolate it to get the y-intercept and the x-intercept, and from that we can determine 1 by Vmax and 1 by Km. So, we can get Vmax and Km. So, why I am showing you this? Because these plots will be very important or very useful to determine which type of inhibitor we are dealing with, right. So what is the first type of inhibition? The first type of inhibition that I am going to discuss is called competitive inhibition. So let's look at this picture. This is our enzyme and this is the active site. This is where the substrate binds and then the reaction will happen, right? The inhibitor looks very similar to the substrate and binds to the same active site. So if it binds there, it will, no further reaction happens, right? So you can see that the substrate and the inhibitor, they are competing for the same active site. So if the substrate is bound, the inhibitor cannot bind, or if the inhibitor is bound, the substrate cannot bind, right? So that is why it's called a competitive inhibition or a competitive inhibitor. And we can write the equation like this: E plus S gives you ES, gives you E plus P. This is our standard Michaelis-Menten kinetics. But the enzyme, free enzyme can also bind the inhibitor and form the enzyme inhibitor complex, and this is given by this Ki or the inhibitor constant. So this is basically the Kd or the dissociation constant of the enzyme inhibitor complex. So now we have to update our Michaelis-Menten equation. So this is the Michaelis-Menten kinetics and this is the kinetics of the enzyme inhibition. So now we have this additional parameter where Ki equals to K minus 3 over K3. So again you can think of this as the dissociation constant of the inhibitor, right? So we have this and we have ES like this. So now we have to update all of this because the total enzyme will be equal to free enzyme plus enzyme bound to the substrate plus enzyme bound to the inhibitor, right? So if we update it like this, we can actually solve for EI, EI is this, and we can solve for ES, ES is this. So we can replace K minus 1 plus K2 by K1 as Km, right? So we can plug in all of this and we will get this form. So we are solving, so we are plugging ES by this and EI by this. So we get this form where the free enzyme concentration equals to total enzyme concentration divided by this, right? And we know the substrate and the inhibitor concentrations. So again, if we manipulate this, so V naught equals to this K2 by Km times ES times S. We know what is E from the previous equation, so we plug in it here and then we do some more manipulation. So what we do is we multiply this Km here, so this becomes, so we multiply it by Km by S. So this S we bring it here, so it becomes Km by S. So this becomes Km by S like this. This cancels out, so it becomes 1 and this becomes this. So now if we take Km by S as common, then it becomes 1 plus Km by S times 1 plus I by Ki. So this I by Ki is left. This 1 plus I by Ki, this is what we call alpha, okay. So this is, you can call it an inhibition constant or some parameter, right. So alpha value will increase if we increase our inhibitor constant, right. So this is alpha and that is multiplied by this. So our final form of the Michaelis-Menten equation looks like this. So Vmax times S divided by S plus alpha times Km, right, where alpha is this. Now you can compare it with the original equation which looked like this. So all we have done is we have replaced this Km by alpha Km, right, where alpha is given by this. So if the inhibitor is 0, then alpha equals to 1. So this equation goes back to this form. But if the inhibitor, there is some value, let's say inhibitor I equals to Ki, then this becomes 1. So alpha will be equal to 2. So alpha will have a value of 2, right. So the effective Michaelis constant will increase, okay. So what this equation tells you that Vmax will not change, but the Km will change, and that is expected because if I go back to this, both I and S are competing for the same active site, right. So if I have some value of I, if I increase my substrate concentration more and more and more, they will outcompete I. So I can actually drive the reaction more towards this direction than this direction, right. So if I have a large amount of substrate compared to the inhibitor, I can drive this reaction in this direction, right. So I can essentially saturate the enzyme by binding all the enzyme by the substrate. So I can actually reach the Vmax value. So Vmax is not going to change, but I will need more substrate to reach that Vmax, right? Because I have to compete out I. So, my Km increases. And that we can see if we plot this, the Michaelis-Menten equation into this, where we are plotting V naught versus S. So, this black line is the uninhibited plot for the uninhibited reaction, right. So, for the same enzyme concentration, if we add a little bit of inhibitor, you can see the line goes like this. So, you see that it will eventually reach the same Vmax, right. If I increase the inhibitor concentration more, it looks like this. So, again it will eventually reach the same Vmax, but after a much higher concentration of substrate. What happens to Km? So, if this is my Vmax, this is my Vmax by 2, right. So, for the uninhibited reaction, my Km is here, but for a little bit of inhibitor, you can see the Km increases. If I increase the inhibitor, my Km increases further, because my alpha value is increasing, right. So, to understand the inhibition, this type of plot is fine, but the Lineweaver-Burke plot is even more convenient. So, instead of plotting it like that, if you plot it in this linearized fashion, then no inhibitor is this. So, my alpha equals to 1. So, inhibitor I equals to 0. So, this is 0. So, my alpha equals to 1, right. So, this alpha equals to 1 line represents no inhibitor, you see. So, this is where I have Vmax and this is where I get Km, right. So, if I increase my inhibitor, let us say I equals to Ki, then this becomes 1. So, alpha becomes 2. You see it goes to the same y-intercept, which means my Vmax is not changing, but now my Km is increasing. So, if Km increases, 1 by Km will decrease, right. If I equals to 2 Ki, then this becomes 2. So, alpha becomes 3. So, it further increases. Vmax still remains the same, but Km increases, right. So, my slope keeps on increasing because my alpha Km is increasing, Vmax remains constant. So, if we have some inhibitor, we do not know where it binds, we do not know how it binds, right. But if we do this simple kinetic experiment and we plot the Lineweaver-Burke plot, if we see our Vmax is not changing, but the slope is increasing, we can tell that this inhibitor is a competitive inhibitor, which means that it is going to bind exactly in the same active site as the substrate. So, there are many examples. One example is shown here. So, this is an HIV protease. So, in the HIV virus, it produces this protease and the substrate binds here. So, there are these different types of peptides which have been designed which inhibit this enzyme, and you can see that atazanavir, which is this one, binds exactly in the same place as the substrate. So, it is a competitive inhibitor because when it binds, it will not allow the substrate to bind, right. So, saquinavir is another peptide and it is also a competitive inhibitor. So, it will also bind in the same place in the same active site as the substrate. Another type of inhibition is uncompetitive inhibition. So, what happens in this case? So, this is our standard equation, enzyme plus substrate gives you the complex which gives you the product. The inhibitor, instead of binding the free enzyme, it binds the enzyme substrate complex and forms this ESI complex, right. So, the substrate binds the enzyme and this can go and form the product. However, if the inhibitor is there, it will bind this and it will form this complex and it inhibits the enzyme, right. So, you can see that somehow binding of the substrate, it either induces some change in the enzyme when the inhibitor can bind, or the substrate binding itself creates a pocket where the inhibitor binds, right. So, the substrate binding actually facilitates the binding of the inhibitor. Here you can actually see that by increasing the substrate concentration, I cannot drive this reaction back, right. So, in this case we can tell that the enzymes which form like this, they will not be easily converted to this unless this inhibitor diffuses out by itself, right. So, I will never gain 100% enzyme substrate complex, right. So, I will never gain the Vmax value. So, here my Vmax is going to decrease and that is what we will see. So, if I do the math in the same manner, right. So, where Ki, So it is Ki dash is defined as this. So I will define alpha dash as 1 plus I by Ki, and I will get this equation. So in this case, instead of alpha being multiplied to Km, it gets multiplied to this substrate. So this is alpha dash, right? And this is your uninhibited equation. So if I flip it, then it will look like this. So instead of alpha getting multiplied here, my alpha dash goes here, right? So if I go to my standard Michaelis-Menten plot, how will it look? This is my Vmax, which is uninhibited. If I add some inhibitor, my Vmax actually goes down. So this red line is the Vmax. So this red line is the Vmax. So this is my Vmax by 2. And if I go to the intercept, my Km will be somewhere here, right? So actually the Km decreases. Okay. The Vmax decreases, but the Km also decreases. But you can see that in this particular plot, it will be very difficult to determine Km. If I increase my inhibitor constant even further, the Vmax goes down even further. So my Km will, Vmax by 2 will be somewhere here, and my Km will be even somewhere here. So this will become really difficult to estimate what is Km. But if we go to our linearized plot, so the equation is given by this, you can see that Km over Vmax has not changed, right? So even though my effective Vmax has changed, so the y-intercept is changing, the effective Vmax is changing, but the slope has not changed, which means that for no inhibitor, if it looks like this, as I keep on increasing my inhibitor concentration, the line will remain parallel to this. So this is a very good characteristic of uncompetitive inhibition. So if you see something like this, you can tell that my inhibitor is binding to the enzyme substrate complex. Okay. So one example of that is this particular inhibitor, which inhibits catechol O-methyl transferase. So again, this is the enzyme. You can see it is all alpha helical. No, sorry, there are some beta strands in the back side. This is the substrate, right? So once the substrate binds, it creates this pocket where the inhibitor binds, right? So inhibitor actually interacts with this magnesium ion, which is interacting with the substrate, and then it is also interacting with this residues of the enzyme. So binding of the substrate is important, because then only this pocket is created where this inhibitor is binding. So it binds to this enzyme substrate complex. The last one that I am going to discuss is called mixed inhibition. So in this case, what we get is the inhibitor can bind the free enzyme, it can also bind the enzyme substrate complex, and they can also interchange, right? So in this case, you will see that I will get an alpha for this, I will get an alpha dash for this, and my Lineweaver-Burke or my Michaelis-Menten equation will be updated, which is basically a combination of the previous two, competitive and uncompetitive. So alpha gets multiplied to Km and alpha dash gets multiplied to the substrate, and these two are defined here, where K and Ki dash are defined here. So this is exactly the same as before. So if we linearize this equation, it will look like this. So my Km is getting multiplied by alpha, and my Vmax, 1 by Vmax gets multiplied by alpha dash, right? So here my slope will increase and my y-intercept will also change. So you see that this is not here, it has shifted, and how it will look will depend on the combination of the relative values of alpha and alpha dash, right? So if you find something like this, then you can sort of predict, or you can estimate that your inhibitor is following a more complex mechanism where it is mixed inhibition. Okay. So these are all reversible inhibitors. We also have inhibitors which are irreversible inhibitors, and here I am giving you some examples of irreversible inhibitors. So irreversible inhibitors, they permanently bind the enzyme. So they kill the enzyme permanently because they form covalent bond with the enzyme. Penicillin and all the penicillin class of molecules, so the related beta lactams, are antibiotics. So they irreversibly inhibit the bacterial transpeptidase enzyme. So transpeptidase enzyme is important for synthesis of bacterial cell walls. So if we can inhibit this enzyme, we can prevent the formation of the cell wall, and that will lead to the cell lysis and death of bacteria. So, penicillin is a irreversible inhibitor. Another inhibitor is 5-fluorouracil, and we are going to look at the structure and of the enzyme and this inhibitor in the next slide. So, this gets metabolized into a compound. So, this is not the inhibitor. Once a patient takes this molecule, it gets metabolized into a compound which forms covalent complex with thymidylate synthase. So, this is the molecule which helps in the synthesis of thymine, right? And it binds this irreversibly, which means that it will actually interfere with the formation of DNA and hence cell division, right. So, 5-fluorouracil is used to treat different forms of solid tumors. Another molecule is allopurinol. So, this is a very common inhibitor. So, it gets metabolized into oxypurinol, which inhibits xanthine oxidase. So, what is this enzyme? This enzyme is involved in the production of uric acid, right. Now, there are people who have, so like again in older people where certain enzymes do not work or something goes wrong. So, uric acid is increased, and then that uric acid gets deposited in different joints in the toes, and then that causes pain in those regions, right. So, those are called gout attacks. Now, if we can reduce the level of uric acid, we can actually treat patients who have gout attacks, and allopurinol is an inhibitor which inhibits xanthine oxidase and prevents the production of uric acid. Okay. So, let us look at this 5-fluorouracil, right. So, this is the enzyme which is thymidylate synthase, and the the inhibitor is bound here, and the enzyme is also bound to methylene tetrahydrofluorate, right. So, tetrahydrofluorate is the one which will ultimately donate this methyl group to uracil and result in the formation of thymine, right. So, that one of the substrates is bound here. So, it is actually a cofactor, and we have the inhibitor which is bound here. So, if we zoom into this region, it looks like this, right. And you can see that it is forming a covalent bond with this cysteine 146, right. So, once this is forming a covalent bond, so it is bound to this, it is also bound to this cofactor, and these two will permanently remain in the active site of the protein. So, this enzyme molecule is, for all practical purposes, it is inactivated, right. So, that is why these are called irreversible inhibitors, because these inhibitors, they will go to the active site, they will use the same reaction mechanism as the enzyme uses, and it forms a covalent bond. So, once this covalent bond is formed, no further reaction happens, right. And since no further reaction happens, this enzyme is permanently, so this inhibitor is permanently bound to the enzyme active site. Okay. So, again these are some of the books that you can follow. You can go through Leninger's Principle of Biochemistry, and you can also go through Molecules of Life. So, these are the two primary books that I have been using. So, you can but you can use any of these books. Okay. Thank you for now.